Yes, hello everybody. This is Franz Schäfer speaking from Heidelberg, Germany. So good afternoon, good morning, good evening, wherever you are listening from. So welcome to this ERKNet webinar. We have this announcement to make in case you have not heard about the recent calls by the European Joint Program for Rare Diseases. If you are a member of an ERN, ERKNet, or another ERN, or affiliated partners, you are entitled to apply for short-term research fellowships up to 3 months. And you can also apply to for funding for scientific workshops. So if you're interested in one of these programs, you can check at the EJPRD website to see how to apply. All right. So today we have a very distinguished speaker who is John Sayer. John is a clinical professor of renal medicine and a consultant nephrologist at Newcastle upon Tyne hospitals. His clinical interest in inherited kidney diseases inspired his research program in recent renal genetics. So within the Institute of Genetic Medicine, his laboratory group studies the genetics of kidney development, cystic kidney disease, and ciliopathies, and also renal stone disease. This research utilizes physiological and genetic techniques, including cellular, murine, and zebrafish models. Within the Newcastle hospitals, he is a clinical lead for the renal genetics service and runs several regional MDT clinics for inherited disorders. Nationally, he leads rare disease groups for inherited kidney diseases and is a clinical champion for genomic medicine. So, John, the cyber floor is yours.
Great, thank you, Franz, and thank you to everyone that's joining this webinar. So my remit for the next 35 minutes is to talk to you about the molecular genetics underlying Joubert syndrome. I'm going to talk in general terms about clinical aspects of the disease, and then I'm going to show you some basic science investigations that we've been doing in Newcastle. I'm going to try and talk clearly and slowly so that you can follow me. So, as Franz mentioned, the work I'm doing is in Newcastle upon Tyne. This is in the northeast of England. And around 10 years ago now, we set up this combined family renal genetics clinic. I'm an adult nephrologist, but my aspiration, I guess, was always to be a pediatric doctor. I didn't succeed in that. I loved the kids, I didn't like the parents so much. But now I work with a pediatric nephrologist and myself, an adult nephrologist and a clinical geneticist, and we deliver this MDT style clinic where we see whole families together to investigate their genetic diseases. And so the remit of the clinic is to see families, provide them with a genetic diagnosis if there's one to be had, and then cascade screening and counselling to family members that may be at risk of inheriting the same disease. So people do travel far and wide actually to come to this clinic, and we get referrals from all sorts of different specialties, mostly actually from pediatric nephrology, but also from our colleagues in surgery, primary care, etc. And people travel from one side of the country to the other to come and see us, and I'm very privileged that people are willing to travel to see us in our clinic. And if we did a review recently to say, okay, well, what are we seeing in this clinic? And you can see that this pie chart over here shows that familial hematuria was a common reason for being referred to the clinic, as well as cystic kidney disease, and the rest of the conditions are rare syndromes, I guess. In terms of genetic diagnosis, these were similarly, we found mutations in the collagen genes underlying familial hematuриas, but also a variety of different genes accounting for cystic kidney disease phenotypes and other inherited disorders. So what are the main benefits of looking at a patient in terms of pursuing a molecular genetic diagnosis? Well, it allows us to give the family a common message. They're not seeing multiple different specialties and multiple appointments. we can be focused on providing a management plan that's specific to their condition. And most of all, I enjoy the clinic because of its research opportunities, actually, if I'm honest. And so, you know, my theory is that any interaction with a patient with a kidney disease provides, one, a learning opportunity, but two, a research opportunity. We know these conditions are rare, and so each family represents a unique opportunity to investigate kidney disease further. So within this clinic setting, we've traditionally collected DNA samples for storage and analysis through panel sequencing, but now we're finding ourselves doing whole exome or whole genome sequencing much more often. And we've also started collecting urine samples from patients with the specific aim to culture their urine-derived renal epithelial cells, and I'll talk to you more about that. Actually, regarding urine, urine is like the oldest medical test in history. People have been writing on clay tablets about urine and its properties for thousands and thousands of years, and the start of laboratory medicine was the study of urine. In fact, before stethoscopes were invented, the mark of being a physician was carrying one of these round bottom flasks called a matula for the specific examination of urine, and people used to gaze at the urine and decide what was wrong with the patient based on the clarity and colour of the urine. And so these ancient sort of tables describing different colours of urine and what this could mean in terms of the patient's phenotype. And it also was open to charlatans pretending to be physicians and pretending to know what's wrong with the patient due to the colour of their urine. The point I'm making here is that, you know, urine acts as a phenotype, and the key to understanding rare diseases is accurate phenotyping to enable us to understand the underlying disease. So this is a diagram of what we do. We take a patient who produces a urine sample. The urine sample is the best sort of second void of the day, clean catch. We take the samples to the laboratory on ice, we spin them down, and we get a pellet of cells which we then plate on here on these 12-well plates. And in around 7 to 14 days time, we get the cells growing as a monolayer, as epithelial cell layer, as a culture. And so this becomes a very powerful way of studying patients with kidney disease. From the cells, we can obtain DNA and do genetic studies, as you know, in comparison to the blood. We can do transcriptomic studies, isolating RNA from the cells. A lot of genes actually don't express their kidney transcripts in white blood cells, and so this offers like a liquid biopsy of kidney tissue that we can do transcriptomic studies on, and we've done this. You could also isolate protein, do proteomic studies, immunofluorescence, EM, and something that we've begun to be doing is taking the urine-derived renal epithelial cells to iPS cells, and then differentiating the iPS cells back to kidney or brain or eye to make organoids. And this is an area of rapid growth. And we can also culture these renal epithelial cells in 3D, and I'll show you some data on that. So my work studies ciliopathies, so I need to tell you what a cilia is if you don't know, or remind you. So the cilia is these antennae that stick up from the cell surface and project. So renal cilia are shown here on the left, projecting into a tubule, and literally they're sort of sensing their environment about the cell. The cilia is thought to tell the cell what's left and what's right, what's top and what's bottom, and there are proteins decorated upon the cilia that sense the environment, and so there are odorant receptors and other receptors on the primary cilium in the kidney that literally are smelling or tasting the urine as it passes by. They may have also mechanosensory properties, although that's an area of debate. So the primary cilium, as I mentioned, sticks up from the cell surface like a flagpole. It's derived at the base from a basal body and then extends outwards. And this zone between the basal body and the cilium is called the transition zone, and that's an important area which decides in this privileged membrane, the ciliary membrane, what goes in and what goes out of the cilium. Shown here, if you can see, is a movie of what we call intraflagellar transport, in that this is a chlamydomonas cilium, but it represents what's happening in mammalian cilium as well, in that things are traveling up and down the cilium all the time. And it's not just a conveyor belt. If you watch the video quite closely, you can see the velocity of things going up the cilium is very different to the velocity of things coming down. So there's one set of motors that propel proteins up the cilium and a different set of motors that propel things down. So the cilium is highly complex in this regard. And there's now a link, well, a link was established some time ago actually, between cystic kidney diseases and proteins that are expressed in the cilium. So if you take pretty much every gene that causes cystic kidney disease, and shown here is PKD1 and PKD2, their protein products are expressed in the primary cilium. So mutations in genes that cause cystic kidney disease express their protein products in the primary cilium. And this has become the paradigm really for renal ciliopathies. And as you can see on this diagram, annotated here is polycystin 1, polycystin 2, other proteins called cystin and polaris. And mutations in any of these genes encoding these proteins cause cystic kidney disease phenotypes. So this link between abnormal cilia and cystic kidney disease is well established. Now moving on to nephronophthiasis. So this is a tricky word that no one can say right. It can be pronounced nephronophthisis or nephronophthiasis, or MPHP, which is easier. But this is an autosomal recessive disorder secondary to defects in primary cilia. And this disorder typically gives end-stage kidney disease between the first 3 decades of life. The symptomology is of polyuria, polydipsia, sometimes severe anemia, growth retardation. And the pathology is very different from polycystic kidney disease in that the cysts are within the kidney at the corticomedullary junction, and they don't distend the kidney, make the kidney enlarge. Typically the kidney is a normal size, and on ultrasound scan there may be loss of what we call corticomedullary differentiation, so the kidney starts to look homogeneous under ultrasound. Under histology, there's tubular basement membrane disruption, tubular atrophy, microcyst formation, and lots of interstitial fibrosis and scarring. And actually, it's one of the most frequent genetic causes of end-stage kidney disease in the first 3 decades of life. You can sort of label a nephronophthiasis by its different onsets of presentation. You can call it infantile, juvenile, or adolescent. Juvenile is a typical presentation, but the infantile cases are noteworthy in that they can present with enlarged cystic kidneys, a bit like ARPKD, and we'll come back to that. The major thing about nephronophthiasis is its extrarenal manifestations that include, in up to 20% of cases, eye disease in the form of retinitis pigmentosa and retinal dystrophy, an eye disorder called oculomotor apraxia, and Joubert syndrome, which is an abnormality of the cerebellum. So shown here is what's called the molar tooth sign. So this is an MRI scan of the brain showing the molar tooth, which is an elongation really of the cerebellar peduncles and a loss of the cerebellar vermis that gives this tooth-like appearance. There can be severe manifestations in the brain, including encephalocele, which can be incompatible with life. I mentioned the retinitis pigmentosa, but also there can be associations with skeletal anomalies, scoliosis, heart defects, including situs inversus, and liver fibrosis. So because the disease is related to primary cilia, it's not unreasonable that these extrarenal manifestations are quite common. The histology, just to remind you, this is normal kidney with back-to-back tubules. This is a kidney affected by nephronophthiasis. You can see it's scarred. There's basement membrane thickening and thinning. This is not pathognomonic for nephronophthiasis, but typical of an end-stage kidney. And you can see that some of the tubules are dilated, and you may see more dramatic corticomedullary cysts than shown here. What genes can cause nephronophthisis? Well, there's now a list of 18 or 19 genes shown here, and really there isn't really a pattern of what's been identified, other than, as I've already mentioned, the protein products of all these genes seem to go to the primary cilium. These are the genes associated with infantile presentations of quite polycystic kidneys. Inversin was the first gene to be described with this, but MPHP3, initially identified as an adolescent form, is actually the most common genetic form of infantile nephronophthisis, and NEC8 and SEP83 have also been shown to be associated with infantile presentations. And you can go in OMIN and browse these lists of genes that cause nephronophthisis. I haven't yet mentioned Joubert syndrome. So Joubert syndrome is the triad of brain, eye, and kidney phenotypes. So the molar tooth sign in the brain, the retinitis pigmentosa in the eye, and the nephronophthisis in the kidney. And remarkably, there are 35 causes now of Joubert syndrome. And again, OMIN lists them as a phenotypic series here. You don't always have to have the kidney phenotype with Joubert syndrome. About a third of cases have a renal phenotype. You can have brain or brain and eye limited phenotypes with Joubert. So one of the most fascinating things about ciliopathies is this huge spectrum of clinical phenotypes. On the left side, the green side, you have the so-called milder phenotypes, although kidney failure is never mild. Nephronophthisis, Senior-Loken syndrome, which is nephronophthisis plus retinal degeneration, and Bartter-Biedl type phenotypes. In the middle is Joubert syndrome, so the brain and eye type phenotypes. And on the right is the severe lethal syndromes called Meckel syndrome or Meckel-Gruber syndrome, and severe skeletal ciliopathies like Jeune syndrome, where there's short rib, thoracic dystrophy, and other problems. And one of my clinical fellows in the laboratory, Shalabh Srivastava, has reviewed all the genetic causes of nephronophthisis recently and provides information about all these causes. It's worth a look at that paper. So now that's enough from me. I'm inviting you to have a question just to break this talk up a bit. So the quick question is, the following type of genetic investigations has allowed the rapid identification of novel genes for MPHP? So I didn't specifically mention this, so this is, intuition, I guess, but could it be Sanger sequencing, linkage studies, panel sequencing, whole exome sequencing, or array CGH? So I invite you to respond. Okie dokie.
Okay. Maybe wait for a little bit longer. 3/4 of people have chosen their answers by now.
Okay, good.
Okay, anybody else to vote? Yeah.
Okay, so you're right. So that's the audience whole exome sequencing. So Sanger sequencing is the gold standard for describing variants, but you're not going to find a novel variant by Sanger sequencing a gene because you don't know where the gene mutation is. Linkage studies traditionally have been a way of finding new genes, and they're still useful in combination with other studies. Panel sequencing, so a panel sequence takes a group of genes, known gene disorders, and sequences them by next generation sequencing. So you're never going to identify a novel cause of Joubert syndrome or MPHP or whatever you're looking for by a panel. And whole exome and whole genome sequencing to identify novel genetic causes. Actually, I work with Friedhelm Hildebrandt in Michigan, and his combination of whole exome sequencing and linkage analysis and homozygosity studies was very powerful to find new genes. So well done for getting the answer right. I think we have a second question now. So which is the most common cause of MPHP? Select one of the following. Is it adolescent MPHP3, whole gene deletion of MPHP1, infantile INV mutations, MPHP4 with retinal dystrophy, and RP-GRIP1 with Kogan motor apraxia?
Okay, 60%. Anybody else to vote? Okay.
Okay, so that's an interesting split. So the answer is whole gene deletion of MPHP1. So MPHP1 is the most common form of nephronophthisis altogether. So we can solve about 70% of cases genetically by looking at all the 19 different genes. And MPHP1 mutations would account for about a third of those cases. And within MPHP1 mutations, the whole gene deletion is very common. Around 25% of MPHP1 deletions will be whole gene deletion. So that can occur homozygously or compound heterozygously with another mutation. MPHP3, I mentioned this as a common cause of both adolescent and infantile nephronophthisis, but it remains a very rare cause of nephronophthisis in total. INV mutations are exceedingly rare, and there's only a handful of families with these mutations. Okay, so time to move on. Okay, so now I'm going to talk about some basic science. So you've had the background introduction, but this is a family that have agreed to share their data with us. So I'm going to talk about these two children, Ben and Emma, and their diagnosis of Joubert syndrome. So Emma, mom had a normal pregnancy with Emma, had a normal delivery, was sent home from hospital, said she had a normal, healthy baby. And then mom noticed that the child wasn't following her around, her eyes weren't following her around the room by the age of 6 months. And then she developed seizures, and it was noted that she was developmentally delayed. That led to a brain scan, which showed a molar tooth sign, MTS. And then to top it all, poor Emma developed renal failure and reached end-stage renal failure before her 2nd year of life. So a very severe phenotype, actually. That's her brain scan on the left showing a molar tooth, not a classical one, but suggestive. And this is her kidneys scan showing some cysts. When Ben, her little brother, was born, mom knew what to look for. Again, normal, uneventful pregnancy, but mom noticed nystagmus and eye problems at a very early age, and poor Ben was blind by 2 months of age. He had developmental delay, scoliosis of the spine, and was developing, like his sister, CKD. He had polyuria and polydipsia. That's his renal ultrasound scan showing some cystic change within the kidney. And this family was found to have compound heterozygous changes in CEP290 by panel sequencing. And here I've drawn a picture of the exons of the gene. This is a huge gene, actually, 54 exons with 54 coding exons, the start codons in exon 2. And the mutations are about halfway along the protein. The protein's illustrated here with the yellow shows core domains, and there's various other very important domains. One of the mutations is a missense, the other looks like one of the mutations is a splice site, a frame shift, the other looks like a missense, but actually it loses a splice site. So you would predict this set of mutations would lead to either a very truncated protein shown at the bottom or nonsense mediated decay and no protein. So I alluded to the transition zone before, but CEP290, the encoded protein, sits at the transition zone, which is the base of the cilium. and regulates what goes in and what goes out of the cilium. And one of the fundamental questions about CEP290 and its mutations is, why does it cause disease, and why does it cause this huge spectrum of diseases? The weird thing about CEP290 is that it can cause nephrophthisis on its own, also Joubert syndrome, but also the very severe syndrome that I mentioned, Meckel-Gruber syndrome, leading to perinatal death. And it also may cause Bartter-Biedl type syndromes. If you take all the other genes involved in nephrophthisis, and many are listed on this busy slide, you can see that the phenotypes are all overlapping, and some genetic causes cause some phenotypes and some don't. So the million-dollar question is, why? One, why does CEP290 cause this diverse range of phenotypes? And two, why, you know, all the other different phenotypes specific to specific genes? So that brings us on to another question. So mutations in the following genes has led to BBS type phenotypes. So is it MPHP1, CEP120, MKS1, CEP290, or NEC8? Okay. Okay, so everyone got the right answer. So CEP290 is associated with BBS type phenotypes. The other genes listed there haven't been to date. There's another gene called CEP164 that seems to be associated with BBS phenotypes as well. Okay, so we'll move on. So I was interested in studying this family a bit more, so we took urine from Ben and took it to the lab. You can see initially the cells have lots of squamous cells amongst them, but after 7 to 10 days you get this monolayer, which is essentially a set of renal epithelial cells. We know it's a mixed population, but we select out for collecting ducts actually, and these seem to be the most robust cells that can grow. And the idea is that we grow these cells either in 2D or 3D to study the cilia structures. We can take cells from any regular control and grow them in 3D actually, and they form these spheroid structures. So you can see in white the cilia poking into the lumen of this structure, which is analogous to a football. And when we grew cells from this child with CEP290 mutations and Joubert syndrome, he was unable to form these 3D structures very well. So we picture a deflated football with loss of primary cilium and loss of lumen, and basically the cells are unable to coordinate themselves into forming a 3D structure. So the next question was, could we do anything about this situation? And a long story short is we worked with animal models to show that there was a defect in the hedgehog signaling pathway, which is a ciliary signaling pathway. And so we thought, well, maybe we can upregulate this pathway and rescue the defect. And so this is exactly what we did with these cells from Ben. And we were able, using a drug called puremorphamine, which is an agonist of the hedgehog signaling pathway, rescue the deflated spheroids into pretty much normal looking spheroids with rescue of the primary cilium. So we were quite excited by this, and we proceeded to validate these experiments in a number of models, but the puremorphamine hedgehog agonist seemed to provide a robust rescue, not possibly to normal wild type levels, but towards wild type levels. The next thing we did was to look at the cells from Ben on a 2D manner, and we noted a couple of things. One was the cells were abnormally long and seemed to have a bulbous tip at the cilium, so there were structural abnormalities that were going on with the functional change in hedgehog that we saw. And so the next question was, does treatment with puremorphamine rescue the ciliary phenotype just like it rescued the spheroid phenotype? And amazingly, we did show that these long, tortuous and sometimes bulbous cilia were rescued into normal-looking cilia with this treatment. So pure morphamine is toxic and could cause glioblastomas. So what we're doing now is looking for alternative therapeutics that can manipulate this pathway without having the same toxicity as pure morphamine. You might say, well, these primary cilia that you're culturing in cells from urine, this might all be an artifact. And so this was work done from Sophie Saunier's group in Paris. They had a very precious couple of patients that had CEP290 mutations who'd also undergone a renal biopsy. And amazingly, these patients both showed that they had abnormally long, tortuous cilia pointing into the lumen of cysts within the kidney. So amazingly, what we're replicating with our renal epithelial cells is what's seen in human biopsies of patients with CEP290 mutations. Okay, time for one last question. So clinically, nephronophthisis may typically present with all except loss of urinary concentration ability, secondary enuresis, that's secondary bedwetting, loss of corticomedullary differentiation, nephrotic range proteinuria, and cystic kidney disease. So you're looking for the odd one out, the one that doesn't cause the phenotype. Okay, so you're right. So very rarely does any significant proteinuria happen with nephronophthisis. All the others may occur, and indeed loss of urinary concentration ability can occur as the sort of initial finding, and measuring urinary concentration or urinary osmolality is a useful key clue to whether the patient has nephronophthisis. Okay, so I'm going to tell you about another family now. So this is a family that we call AA, and this young man was 14 years old. You can see that he's wearing glasses, but he attends a sort of special school, has some learning difficulties, but functionally is doing not too badly. And he has the mild cystic kidney disease change and a molar tooth sign on MRI scan. And this chap has Joubert syndrome by definition, and we found a homozygous mutation in CEP290 accounting for his disease. He has progressive kidney failure, but able to give us urine samples whenever we like. So this mutation causes a stop codon in exon 42, shown here, leading to a predicted truncated protein. And so with this chap's urine, we did the same thing as I've shown before, collected a urine sample, grew a renal epithelial culture, and used it as a model to study this disease. We have an antibody just to the C-terminal of CEP290, and this blot shows that there's no protein, at least no full-length protein, present in this man's cells derived from his urine. And amazingly, his cilia on renal epithelial cells showed this wiggly, long, tortuous phenotype, just like we've seen in the other family. And so the question was, is this treatable as well? And so we'd done the pure morphamine trick, but we wanted to try something else. I'm going to skip by that one. So here we show that the mutation is in a single exon 41 that encodes 123 nucleotides. So this is a potentially skippable exon because it's a multiple of 3. And we noted that this exon encodes a discrete coil-core domain, didn't seem vital for structure or function, and therefore we used a technique called antisense oligonucleotides to induce alternate splicing of exon 41 such that this exon is completely missed out. And so when the transcript is read, we no longer see a stop codon in exon 41 because that exon's bypassed, and we get a near full-length protein. And so this is exactly what we did. We designed an oligonucleotide to induce alternate splicing. And when we treated cells from AA with this ASO, we got a rescue of this ultra-long cilia to a normal length cilia. And shown here is the abnormal splicing that's been induced. And shown here on Western blottings, we see a return of near full-length protein in the treated cells, which was quite interesting. And so the key question now is to say, okay, we can rescue the cells, can it rescue the kidney phenotype? Shown here on the right-hand side is actually a return of the CEP290 protein, shown as this white blob, pseudo-colored white, when we treat with the exon skipping therapy. And so we have a mouse model of CEP290 mutations. It's not identical to the patient. It includes a gene trap. So here the alternate splicing was to splice out our gene trap, which we'd engineered into these mice. And amazingly, when we used a targeted antisense oligonucleotide to induce alternate splicing of our gene trap, we saw a rescue of the cystic kidney disease phenotype seen in these animals. And shown here is the rescue of the full-length protein once the gene trap had been alternatively spliced out. And we were able to show both the ciliary rescue from long cilia to normal length cilia, and a rescue of the cystic kidney disease in these mice kidneys. So now we're moving on to work with a mouse that has the exact same mutation as that patient AA, and to explore delivery and dosing strategies of the antisense oligonucleotide to the kidney tissue. These antisense oligonucleotides are injected by an intravenous or subcut injection, and they're taken up by the body, freely filtered at the glomerulus, and end up in the tubules, which is exactly where we want them to be. The key question is, are there other mutations in this rather large gene that are potentially skippable with the same therapy? As I mentioned, there's 54 exons, and 26 seem skippable, although some do encode important functional domains. If we draw a little map of where the mutations are, in green shows where the bar chart shows the number of mutations, green shows whether they're potentially skippable. This is the mutation in our patient AA, but you can see that there's another more common mutation seen in many more probands that's potentially treatable with the same technology. Okay, so I'm running out of time now, so I'm going to skip these ones. I want to conclude by saying that thanks for listening, and remind you that every interaction with a patient is a potential learning opportunity for research as well. Deep phenotyping of patients is important, and we've used the urine-derived kidney cells to phenotype these patient cilia. The urine provides a liquid biopsy of the kidney. It's completely non-invasive, and nearly every patient will agree to it. And the discovery of genes allows us some insights into how this disease and ciliopathies work, and leads us into investigations into therapies. Collaborations and team science are always important, working with rare disease and setting up networks like ERKNet, and in the UK we have a network called Radar, is very important. I don't do the laboratory work on my own, and we're supported by funding from all these agencies. So thank you for listening, and I'm happy to have some questions.
Yeah, thank you very much. This was a very impressive lecture. So if you want to ask questions to John, please write them up, and I can read them for you. Okay. So while people are thinking about questions, may I start? So I was actually very intrigued by these two siblings with the same disease, and I was thinking in your multidisciplinary clinic, are you offering, in the UK, are you offering pre-implantation diagnostics to such families?
Yeah, so for this family in particular, we did go down that route actually, and we went nearly all the way, and then other things happened. So PGG is available for the genes that cause Joubert syndrome, and so yeah, if this family wanted another child, they were prepared to go down that route. Having two pretty severely disabled children was, you know, enough for this family at this particular time.
Mm-hmm. The other question is about the exon skipping therapy. I mean, this is, of course, a very intriguing therapeutic approach. I know it has been tested in some clinical trials already in, I think, some neuromuscular disorders. Can you give us an overview on what has been the experience with exon skipping therapies in clinical research so far in other diseases?
Yeah, so Duchenne muscular dystrophy is the most notable example of exon skipping, and so dystrophin is a very large gene, and you can, using exon skipping, turn the dystrophin mutation from a Duchenne type mutation into a Becker's, so a milder form of muscular dystrophy with good effects. And so they've been using in clinical trials intramuscular injections of antisense oligonucleotides for this specific effect. I think the huge advantage of using antisense oligonucleotides for exon skipping in the kidney is, as I mentioned, the kidney seems to freely filter these, and they end up in the tubules. They're in the primary filtrate. They're endocytosed by the tubules almost by accident, really. And so you're getting targeting of the kidney tissue, the tubules in particular, that you can use to your advantage. I think some of the problems with treating dystrophy is the dosing required to get delivery to the large muscles that are involved. But for kidney disease, it remains very attractive.
Mm-hmm. So I think these oligonucleotides are quite stable molecules. So how often would these have to be administered for a clinical effect? What would be your expectation?
So we're thinking, so in our animal models, we've seen after a single dose, the effect lasts for over 10 days, actually. We think that's part of the reason that is CEP290 in particular has a slow turnover. Once it's made, it seems to stay in its location at the transition zone for a long time before it's required to be turned over. So I imagine clinical trials would involve some dosing of fortnightly intervals.
Okay.
You think that would be invasive? There's lots of other therapies that require, more invasive regimes.
Absolutely. All right. So I think there's no more questions coming in. So I thank you very much, John, for this lecture, and everybody for listening. And you can see our next webinars. We will have an IPNA clinical practice webinar on growth hormone treatment in CKD children on the 5th of March. And then there will be an educational webinar on CACUT by Carl Bates on the 10th of March. And then the next advanced webinar on rare kidney disorders will be held on the 24th of March on the topic of steroid-resistant nephrotic syndrome in mitochondrial cytopathies. So thanks everybody for listening, and have a nice evening. Bye-bye.
Bye-bye.
Okay.